So Darwin proposed that natural selection was the primary mechanism for change in species over time. He recognized that variation in heritable traits was a prerequisite for evolution, but he didn't know how exactly organisms pass traits on. A few years after, Gregor Mendel proposed a modern inheritance that supported Darwin's ideas, and so basically our modern idea is more of this neo-Darwinistic perspective that incorporates our understanding of both natural selection and inheritance and genetics. So we know now that genetic variation occurs within a population and individual variation occurs in all species that reflects that. Genes can be inherited, but not all changes are heritable. Phenotypes may be influenced by environmental factors, for example, or you may have changes that are not in a gametic cell for a sexually reproducing organisms. So substitutions and insertions and genetic changes, overall, that can lead to evolutionary change. You could look at the amount of heterozygosity, you could look at nucleotide variation, you could look at geographic variation. Basically, variation exists. There's lots of different types, but it doesn't necessarily have to be a disadvantage or an advantage and broader focus. And it could be something that's not genetically controlled at all. The caterpillars down here at the bottom, for example, are the same species. The difference is in what they ate. That conferred different advantages. So those variations may not be physical ones, they may be behavioral ones, but it still is something that evolution can influence. That drive leads us to our sort of modern synthesis of evolutionary understanding. The modern synthesis introduces several changes in how evolutionary processes were conceived, and it proposed the idea that evolution is a change in allele frequencies within a population over time. This is emphasizing this genetic basis of evolution. Overall, there are four forces that are thought to drive this process. You can see them here on the right. Of these, natural selection, by which the best adapted organisms have the highest survival rate, is the only force that make organisms better adapted to their environments. Genetic drift describes random changes in allele frequencies in a population. It's particularly powerful in small populations, as we'll look at in a moment. Gene flow looks at the allele frequency changes due to immigration and emigration, and mutation, which is the weakest evolutionary force, but still critical because all genetic variation originally arises from variation. Overall, then, changes in species occur gradually through the accumulation of small changes. This is at least one of the key ideas, and that's influenced by the prior concepts from geology and gradualism. The large differences that are observed between species involve extensive time periods, looking at that uniformitarianism concept. The modern synthesis is still taking in new information, it's still being revised and changed, so the focus could shift, but these are the major driving forces. Genetic drift, again, random change in frequencies in a population due to chance events, is, like I said, random. You have an existing population, it has a certain allele frequency, but not everything is going to survive. So in this example, we had pink and red and white, but the alleles, not all of them reproduced, only some of them reproduced, leading to a change in the population. And that change can accumulate over time, leading to massive changes in the species. There are considered to be two major types of genetic drifts. When you have small populations that are being selected for, and it really comes down to, did the original population survive the event or not? A bottleneck event is going to be where you have an original population and something is going to cause a very small amount to survive. That bottleneck is going to be a drastic disaster of some kind, leading to some alleles being unrepresented or underrepresented in the survivors. You could also have the founder effect, like colonization, for example. Just because the Quakers or whatever came to America to start a population does not mean that their ancestry, their related relatives didn't stay in England or wherever they started from. Okay. Genetic drift plays an important role in conservation efforts as well. So for example, the greater prairie chicken on the right, they were a wide range species, but they were knocked back to very isolated populations due to a bottleneck event. And we were able to bring the species back from near extinction in the state because they were able to bring in prairie chickens from other states, Kansas and Nebraska, and were able to help the species recover. So genetic drift can be reversed if there are other populations that can bring new alleles in, but that ties into the idea of gene flow. Gene flow looks at breeding, basically, and our modern concept of these difference between a species is that a biological species is one in which the organisms can interbreed. So gene flow, a change in the frequency of traits in a population due to immigration or emigration, is going to be key to that concept. Gene flow tends to be an equalizing force. It can be hard to predict its effects, but it leads to a spread of traits over time, leading to changes in allele frequencies. Alleles transferred by gene flow can affect how well populations are adapted to local environments. It tends to reduce differences between populations. If it's extensive enough, gene flow can actually lead to populations combining into a single population over time. And you can sort of see the overlap here between these two herds of look like caribou. Okay, there's going to be extensive gene flow where the range is overlapped, but not where they are separate. But that still means that traits can spread through this population. And that's true for a variety of organisms and structures and developments. Basically, beneficial alleles can be transferred between populations, and gene flow is an increasingly important agent of evolutionary chains, especially in humans. With increased human mobility, mating is more common between previously isolated populations, leading to an exchange of alleles and reducing genetic differences between human populations. That connects to our sort of mathematical and statistical relationships for determining changes in allele frequency over time. You have your gene pool. That is going to be all the genes, including all of the different alleles of all of the individuals in a population. A population is a group of individuals of the same species that live in the same area and interbreed to produce fertile offspring. So in this example, we have black mice, we have brown mice. Some of them are homozygous, some of them are heterozygous, and that population represents a given frequency. And if that frequency changes over time, then the population is said to have evolved. In this example, we have 100 mice. The gene pool therefore has 200 alleles, and of those 200 alleles, we have 80 that are dominant and 120 that are recessive, and that can factor into your calculations. There is some sort of basic math here. If you look at the frequency of alleles, so we think back to the red and white genes for the red and white flowers, P is typically representative of the dominant allele, Q is recessive. So if we look at all of those alleles, we have an 80% chance of randomly drawing a red allele, a 20% chance of randomly drawing a white allele. The gametes that are going to be produced then have an 80-20 chance. That's going to be the basis of our gene pool, leading to selective change. If you take that and tie that into statistics, you end up with a very basic test called the Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium is a null hypothesis for evolution. If a population is in equilibrium, then it is not changing over time. If you have two traits, two versions, then the dominant and the recessive, P and Q, is equal to 100% of the population. So the frequency of P and the frequency of Q will equal to 1. If you then FOIL that out and look at a diploid organism, you have three possible arrangements. You have homozygous dominant, which is represented by P squared, heterozygous 2PQ, and homozygous recessive Q squared, which cumulatively would represent 100% of the population. If we take that 80-20 split, that means that we have a 64% chance of having a dominant red flower, and then we have the remainders here that you can see. So 0.16 and 0.16, we need to add that up to get our populations. So we have a 64% chance of being dominant-dominant, a 32% chance of being heterozygous, and a 4% chance of being homozygous recessive. So we can see that interrelationship here. But again, it is a null hypothesis. You are not evolving. So that's something to consider. To be in equilibrium, there needs to be no mutations. The gene pool is modified if mutations alter alleles. Therefore, if there's like a deletion or a duplication, then they are evolving. Random mating must be maintained. Why? No sexual selection. Basically, you could say really there's no selection of any type, but the original one just said no natural selection. But now we know that there's frequency dependent and artificial selection. So yeah, no selection of any kind. 4, the population needs to be large so that genetic drift will not impact the population. And then they need to be isolated so that gene flow will not impact the population. Departure from any of these conditions results in evolutionary change. So if you calculate the Hardy-Weinberg equilibrium, if you look at the frequency of the alleles at the beginning and the end, if they are at equilibrium, there will be no statistically significant difference between those values. The rate of evolutionary change in many populations is so slow that they appear to be close to equilibrium, so we can still use this to estimate our changes and variations within structures. Departure doesn't have to be for the whole organism. A population can be evolving at some loci, but yet simultaneously be in equilibrium for others, so you do need to test your ideas. If you are attempting to solve a Hardy-Weinberg equilibrium question, here is a sample problem that you can solve. I would suggest you pause here, try it out for yourself, and then continue the process to compare your answers. I'm assuming at this point you have calculated this. If we are looking at this population, if we have 200 newts and 8 are not poisonous, that is going to be our base population. Okay, so if poisonous is dominant over not being poisonous, then the 8 organisms are going to be the homozygous recessive. So from this, we're going to eventually realize, hopefully, that our base population, our frequencies, P is going to be 0.8 and Q is going to be 0.2 from that value. Okay, if we then break that down into our various comparisons, we realize within this structure, 50 newts are washed downstream after a big storm and colonize a new pond. What do you expect the frequency and number of each genotype to be? Okay, well, I can ply that out. P squared would be 0.64, so 32. Homozygous would be 0.32 or 16, and homozygous recessive would be 0.04 or 2. C, you count the new population of newts and find 21 homozygous, 23 heterozygous, and 6 homozygous. Is that what you expect? I would need to do a chi-square analysis to do that. If it's not, what are the new allele frequencies? Well, we realize within this, if you do your chi-square analysis, that the chi-square value that you get is 14.8, which is a large value. Therefore, you are going to reject the null hypothesis. And we'll try more of these in class, so I hope this makes sense.